My name is Ken Hayworth, and I want to make clear the following disclaimer. The Brain Preservation Foundation is a nonprofit science advocacy organization only. We do not offer preservation services, and we will never offer preservation services. And we are currently not endorsing any other company that may be offering preservation services. The mission of the Brain Preservation Foundation is solely to promote the science and application of brain preservation within the larger scientific and medical communities.
The contribution of the Brain Preservation Foundation is really straightforward. Before, there wasn't any way to preserve synapses for 100 years is what the prize criteria was. And now, thanks to that prize, we have a way to preserve synapses for 100 years. Tremendous progress in making brain banking technology dramatically better.
The Brain Preservation Foundation was founded about 8 years ago when nobody knew that you could preserve a brain. In fact, the major thing that the Brain Preservation Foundation did was put out a challenge prize to both the scientific community and the cryonics community to develop just such a procedure.
For 50 years, neuroscientists had been perfectly preserving tissue the size of a millimeter cubed. And it didn't look like there was any artifacts in there. It looked like everything was preserved. It looked under electron microscope, it looked great. Nobody had tried to scale that up to the size of a whole animal brain so that it would look as good for the whole brain as it did for these little tiny pieces. So we wanted to show that the ultrastructure, which is what you can see down to the level of about a 6 nanometer resolution, the electron microscope, was all there. And so we wanted the prize as a way to increase transparency around what these technologies could do, and also to keep everybody in the larger community that's talking about the futuristic implications of these, keep them honest.
Prizes are a way to bring a community's attention to something with clarity. Here is a hard task. Here's how we define that. Some of the people who were working on the brain preservation prizes probably wouldn't have done that if Ken had not elucidated hard but achievable goals.
Now, our challenge prize was designed with the goal of just showing that the structural connectivity, the synaptic connectivity of the brain could be preserved. This was a long process. There were 2 competitors in this prize, actually 3 methods, because one of the competitor teams had 2 different methods. The initial tissue that was given to us was horrible on all fronts. And so it was more disappointing at first. Unfortunately, the cryonics community has yet to meet that milestone. But one group from the larger scientific community was able to meet that milestone about a year ago.
I've always been interested in how the brain works, how memories works, and that dream just stayed with me, even when I went to MIT, where I imagine I'd be studying neuroscience, studying computer science. And I looked at that technology and I saw that they could do this 2-photon calcium imaging and see some electrical activity, but it was very far away from what you would need to actually scan a brain and actually extract the totality of information that's contained in that brain. So I actually stopped doing the neuroscience and started doing the AI, thinking that the better we understand how AI works, the better we understand the nature of intelligence, and good AIs could potentially design better brain scanning technologies. And it was when I found this Brain Preservation Prize, that just seemed like such a brilliant idea. I wanted to be part of that. Here was a really rigorous neuroscientist, Ken Hayworth, putting together a scientific prize with really clear criteria saying that synapses are known to be absolutely critical for long-term memory storage. And so as a first step towards preserving long-term memories, can we preserve synapses and demonstrate that we can preserve them? And so I started volunteering for the Brain Preservation Foundation, and I was trying to make this cartoon that you could put on YouTube that would describe the two competitors. And so I started researching their techniques really carefully so I could learn everything I could about them to represent them properly. And then as I was looking through them, I thought, well, I think I know how to win this prize. You can kind of sort of combine them. And so I went to the cryobiology company and I said, I think we can win this prize. We do it a bit differently. They set me up with the lab and gave me 2 years of time to do it. And then ended up taking about 9 months.
They developed a protocol, and they were able to show that the ultrastructural details of whole rabbit brains and whole pig brains can be preserved for an indefinite period of time. I was extremely excited that somebody had actually demonstrated that a brain could be preserved. But I'll tell you the moment that I was really excited, because the eventual technique that won was a combination of a technique that every neuroscientist knows and loved and has been using since the 1950s. It's glutaraldehyde perfusion fixation. And so there was no surprise that that could temporarily preserve the structure of the brain at that level. The thing that was surprising is that that technique could be made compatible with long-term cryostorage. I had heard from people that I believed, because they had done some basic experiments in this, that compatibility was impossible, or at least extremely difficult. And when Robert McIntyre and his team developed the relatively straightforward, in hindsight, tricks that were needed to get those two things compatible, I was really excited and jazzed that was possible.
There's a lot of complicated chemical processing you have to do to get tissue ready for electron microscopy. And Hayworth actually had to come to Southern California to witness that the brain was intact when we preserved it, because then we later sliced it to prepare the slices. And we had this marathon session of preparing the slices that took all night. Because I think that it's important to continue to develop and continue to research this brain banking technology and get it to a point where you can demonstrate that it's effective at preserving long-term memories. Right now we've demonstrated that we can preserve structure, and now there's more development work that needs to be done. There's more research that needs to be done.
There is a bit of a misunderstanding about the milestone that was set for the Brain Preservation Prize and actually what the winner of the Brain Preservation Prize can preserve. That technique, yes, it does preserve the structural connectivity of the brain, but because it is essentially glutaraldehyde perfusion, it preserves functional molecular features as well. So if you are saying that the real functional strength of a synapse is determined not by its size, but by its receptor protein complement that is expressed, aldehyde stabilized cryopreservation preserves those ion channel and receptor proteins on the synapses.
What do you actually have there? Do you have memories? Do you have personality? What do you have there, and what can you do with that? You might not be able to interpret it now, all of it, but you'll be able to interpret more and more of it going forward.
First and foremost, I would like to see some additional follow-up studies on the aldehyde stabilized cryopreservation experiments that Robert has run. I'd like to see them validated by other scientific groups. I'd like to see them built out further.
I look back at this now, and I think a lot of neuroscientists can look back at this now and say, of course that would work. Why was anybody thinking that it was impossible to preserve a brain? And that has been the reaction of a lot of my neuroscience colleagues. In retrospect, it seems utterly simple.